Good morning, students. How are you? Hope you all are fine. We continue with the chapter Reproduction in Animals. Now we come to next. After the fertilization, we have done the fertilization in the previous class. Today we will study about the development of embryo. You know that in the fertilization, the one nucleus comes from the male and other from the female. Nucleus from the sperm and nucleus from the ova fertilize to form a single nucleus. As we know that the half of the characteristics in the sexual reproduction comes from the parent, one parent, and other half from the other parent, means half from father and half from mother. And zygote is formed and which has the characteristics of both the parents. Now in the human being, we are studying the development of embryo in the human. In the human being, as we know, the fertilization is internal, and the zygote which is formed in the internal fertilization develop in the mother's womb. You know that the zygote start dividing and it forms a ball-like structure. You can see the fertilized egg, then there is 2-cell stage, 4-cell stage, 8-cell stage, 16-cell, then there is blastocyst. So this is the developmental stages. First of all, the embryo, the zygote develop to form a ball-like structure, and this ball-like structure is attached to the soft spongy cushion which is present in the uterus. Uterus is called the mother's womb. You can see the ball-like structures. It forms, develop, it goes inside the mother's womb. You can see. Now in the uterus, the spongy tissues are present, which is called the endometrium. You know that endometrium is meant for giving the protection to the developing embryo. And this process, when this embryo is attached to the wall of uterus, is called implantation. I am repeating, the development or attachment of the developing embryo to the wall of uterus is called the implantation. Now the developing embryo, it develops inside the uterus in the mother form, and it has blood supply also, and it form a link, it develop a link by which it get the nutrition and blood from the mother. That link is called the umbilical cord. That link is called umbilical cord. Now this umbilical cord is also called simple cord. You have seen, बेटा जी आपने देखा होगा, when a baby born, जब एक बेबी का बर्थ होता है, देयर इज ए कॉर्ड ऑन इट्स नेवल, उसकी नाभि पे एक कॉर्ड होती है, दैट इज कॉल्ड द एम्बिलिकल कॉर्ड. एंड व्हेन द बर्थ अकर्स, जब बेबी का बर्थ होता है, द डॉक्टर कट दैट कॉर्ड. डॉक्टर जो है वो उस कॉर्ड को अलग कर देते हैं, बिकॉज द नाउ द बेबी इज नॉट इनसाइड द यूटेरस, नॉट इन द मदर्स वॉर्म, इट इज आउटसाइड. सो द पॉइंट ऑफ अटैचमेंट ऑफ द कॉर्ड विद द वॉल ऑफ यूटेरस इज कॉल्ड द प्लेसेंटा. The point of attachment of this umbilical cord with the mother's womb is called the placenta. The embryo start drawing its nutrition from the blood through this umbilical cord or the placenta. Now, how the baby develop in the mother's womb? How it happens? You know that by the time when the embryo is developed, I am again showing the stages to you. These are the development stages of the embryo. Now by the time embryo is of 5 weeks old, it may have eyes and being heard. By the time it is of 12 week, the embryo is immature baby and it is called the fetus. It is called the fetus. It takes about 270 to 280 days or approximate 9 months for an embryo to develop into a baby, and this period is called the gestation period. Gestation. This period is called gestation period, and when the fetus is fully developed into the baby, the mother gives birth to the baby. So this is all about the development of embryo inside the mother's womb. Now we come to the next slide, that is the cloning. It's very important, cloning. What is a clone? Clone is the same carbon copy of the parent which we get it by the asexual method of reproduction. That is called the cloning. It is a baby produced asexually from a body cell. It is also called the artificial reproduction. It is asexual multiplication. Now, first of all, the cloning is done in a sheep, and the sheep which is taken the birth on 15 July 1996 was named Dolly. Now, you can see in the diagram there are two sheep. One is the black faced sheep and other is the white faced sheep. Two sheeps are there. अब जो black faced sheep है उसमें से egg लिया. You can see egg is taken from that black faced sheep, and from that egg the nucleus is removed. From that egg the nucleus is removed, and from the white faced sheep a cell is taken. Only a single cell is taken from the any gland, from the mammary gland it is taken, and the nucleus of that is inserted in this. Now the egg is fused with the cell. The egg has the nucleus of the cell of white faced sheep. I am repeating. Two sheep has taken. दो sheep ली, एक black face थी और एक white face sheep थी. जो black face sheep है उससे egg लिया और उस egg में से nucleus को remove किया और उसके अंदर क्या insert किया? The nucleus of the normal cell which is taken from the white sheep. Now this is fused. Now the egg has the nucleus of the cell. Egg fused with the cell or the cell nucleus. Now the embryo is formed. By this the embryo is formed when the fusion occur, and then this embryo is implanted in the black sheep यूटरस. उस एम्ब्रियो को इम्प्लांट किया, बॉडी में डाला किसके? ब्लैक फेस शीप के. एंड आफ्टर दिस इट इज टोटल प्रोसेस ऑफ डेवलपमेंट ऑफ एम्ब्रियो एंड द न्यू शीप विच बॉर्न, जो क्लोन प्रेजेंट हुआ, जो न्यू शीप उसका बर्थ हुआ, इट हैज़ द वाइट फेस. उसका फेस क्या था? वाइट फेस थी. द डॉली वॉज बॉर्न विद अ वाइट फेस एंड करेक्टर्स फ्रॉम द शीप विच हैज़ द न्यूक्लियस. जिसका न्यूक्लियस लिया था उसके करेक्टर्स थे, और उसका डॉली में एक भी करेक्टर नहीं था ब्लैक का. डॉली डज नॉट हैव द ब्लैक फेस शीप करेक्टर. डॉली वॉज हेल्दी एंड ऑन 5th July 1996 it was born, and it died on 14th February 2003 after due to some pulmonary disease. Fine. So beta ji, this is called the cloning. Now